Assalamu alaikum, dear students. Welcome to the course Principle in Animal Life 1. This is lecture number 58. The topic is cellular membranes. Discussing agenda for the topic will be cellular membranes are fluid mosaics of lipids and proteins, the fluidity of membranes, evolution of differences in membrane lipid composition, membrane proteins and their functions, synthesis and sidedness of membranes, membrane structure results in selective permeability, the permeability of the lipid bilayer, and finally the transport proteins. Lecture outcome. After watching and listening this lecture, students will know about the plasma membrane, its structure and functions. Introduction. Cellular membranes are fluid mosaics of lipids and proteins. Lipid bilayer is, of course, the ground substance of the cell membrane, and different proteins are embedded within this lipid bilayer. So lipids and proteins are the staple ingredients of membranes. They are the two principal components of membranes, although carbohydrates are also important. The most abundant lipids in most membranes are special types of lipids called phospholipids. Their ability to form membranes is inherent in their molecular structure. The phospholipids are amphipathic molecules, that is, their two ends show different properties. The head region is hydrophilic and the tails are hydrophobic. So this portion is the head portion of the phospholipid and these are the tails. The head portion is hydrophilic and the tail portions are hydrophobic. So the two lipid bilayers are oriented in this way, where the hydrophilic portion is facing towards aqueous environment, both outside the cell and inside the cell, whereas the hydrophobic portions are present in the middle, facing towards each other. Like membrane lipids, most membrane proteins are amphipathic to the proteins which are embedded in the plasma membrane. They also exhibit that characteristic where one portion which is facing towards the aqueous environment is hydrophilic in nature, whereas the portion which is present between the phospholipid tails is hydrophobic, just like the phospholipid tails. Such proteins can reside in the phospholipid bilayer with their hydrophilic regions protruding outward. This molecular orientation maximizes contact of hydrophilic regions of a protein with water in the cytosol and extracellular fluid, while providing their hydrophobic parts with a non-aqueous environment. So the proteins which are embedded in the plasma membrane are also amphipathic. So the region which is facing outward and the other which is facing inward, both are aqueous environments. So that particular region which is in contact with water is hydrophilic in nature, and that portion, that inner portion which is embedded along with the phospholipid tails is hydrophobic in nature. And in channel proteins, usually there is a channel which helps in the transport of materials. So in channels proteins, this channel is hydrophilic. Now the proteins, of course the phospholipids are the main fabric, and then proteins are embedded in this main fabric at various locations, but the proteins are not randomly distributed in the membrane. However, groups of proteins are often associated in long-lasting specialized patches where they carry out common functions. It is unlike the phospholipids where they not only show the longitudinal movement but also the transverse movement. The membrane proteins are fixed at specific locations, and they carry out different functions. And the distribution is also uneven. In some of the regions of the plasma membrane there will be more proteins embedded, whereas at the other regions there will be less number of proteins embedded. Obviously this presence of proteins in bulk in one area and in lesser amount in other area is related with their function, because the proteins which are present in large amount in a particular area carry out the transport process efficiently, or the molecules which tends to enter the cell are in majority in those portions as compared to other areas where the membrane proteins are in lesser concentration. Now the fluidity of membranes. The plasma membrane is a selective barrier. It is a selective traffic regulator, and it needs to maintain a proper level of fluidity. If it becomes too much fluid, then any kind of material will enter the cell, and obviously it will affect the homeostasis inside the cell. Or if it became rigid and solid, then nothing will enter into the cell, and obviously in that condition the homeostasis inside the cell will also be affected. So there must be some kind of check and balance on the level of fluidity a plasma membrane shows under a given set of conditions. So membranes are not static sheets of molecules locked rigidly in place. A membrane is held together mainly by hydrophobic interactions, which are much weaker than covalent bonds. So most of the lipids and some proteins can shift about sidewise. They can shift their positions sidewise, that is in the plane of the membrane, like partygoers elbowing their way through a crowded room. Very rarely a lipid may also show a flip-flop cross movement. It is rare because it happens only once in a month, switching from one phospholipid layer to the other. The sidewise movement of phospholipids within the membrane is rapid. It occurs rapidly, about 10 raised to the power 7 times per second. and a single phospholipid molecule travels about 2 millimeter in a second, that is equal to the length of a typical bacterium. Proteins are much larger than lipids and move more slowly when they do move. Therefore, in the previous slides, we said that the proteins are somewhat permanent structures that reside at specific locations because their large size hampers their free movement in a lipid bilayer. Many membrane proteins seems to be held immobile by their attachment to the cytoskeleton or to the extracellular matrix. So the proteins which are located in the plasma membrane, these proteins are associated on both sides, on the inner side with the cytoskeleton, on the outer side with the extracellular mass. So because of that association on both sides and because of their larger size, their free movement is hampered. So here is diagrammatic representation of the plasma membrane. You can see here it is a lipid bilayer, that is two layers of phospholipids, but the phospholipids are oriented in such a way that their heads or the hydrophilic portions are facing towards aqueous environment, both outside the cell and inside the cell, and their hydrophobic portions, which are the fatty acid tails, they are oriented on the inside away from water, and these large molecules are of proteins which act as channels for the movement of materials or as transport proteins. In the next slides we will study the function of membrane proteins. Some of the membrane proteins have lipids attached to them, whereas others have carbohydrates attached to them. Carbohydrates can be attached to the phospholipid heads also, and they have function in cell to cell recognition. And there you have the molecule of cholesterol. Cholesterol acts as a fluidity buffer. It prevents the it puts a check on the level of fluidity a plasma membrane will show under a given set of conditions. And there you have these proteins that are associated both on the inside and outside with the cytoskeletal elements, on the inside with the cytoskeleton, on the outside with the extracellular matrix. Some membrane proteins seem to move in a slight, highly directed manner, perhaps driven along cytoskeletal fibers by motor proteins. So some of the proteins which are embedded in the plasma membrane, they show higher degree of mobility because they are associated with the cytoskeletal fibers by motor proteins. A membrane remains fluid as temperature decreases until the phospholipid settle into a closely packed arrangement and the membrane solidifies. Such a bacon grease forms lard, such much as bacon grease forms lard when it cools. So when the temperature decreases, the phospholipid tails they become closely packed with one another, and as a result the membrane solidifies and the permeability of the membrane is decreased. Now the temperature at which a membrane solidifies depends on the types of lipids it is made up of. As the temperature decreases, the membrane remains fluid to a lower temperature if it is rich in phospholipids with unsaturated hydrocarbon tails. So, if unsaturated hydrocarbon tails are present, that is, if a double bond is present or a triple bond is present in a phospholipid tail, then at that particular point this tail is tilted or twisted to a certain extent, and therefore this twisted tail pushes the adjoining phospholipids and creates room and increases the permeability. So the presence of unsaturated fatty acids increases permeability, and the presence of saturated fatty acids decreases permeability, because the tails of saturated fatty acids are straight, and therefore they are closely packed with each other, thereby decreasing the fluidity. Now the unsaturated fatty acids, they have kinks in the tails where double bond are located. Unsaturated hydrocarbon tails cannot pack together as closely as saturated hydrocarbon tails, making the membrane more fluid. And the steroid cholesterol, which is wedged between phospholipid molecules in the plasma membrane of animals, has different effects on membrane fluidity at different temperatures. So the cholesterol molecule which is embedded within the phospholipid tails is a selectivity buffer, fluidity buffer. At relatively high temperature, that is at 37 degrees centigrade, which is the body temperature of humans, for example, cholesterol makes the membrane less fluid by restraining phospholipid movements. So the frequency, not only the presence of saturated and unsaturated tails, but also the movement of the individual phospholipid molecules, they also determines the level of fluidity to a certain extent. So what is the prime role of the cholesterol? The prime role of cholesterol is to decrease the movement of the individual phospholipid molecules, and thereby the fluidity of the membrane is decreased. However, because cholesterol also hinders the close packing, because cholesterol is also present alongside in between the phospholipid tails, so on one side it is decreasing the fluidity by preventing the movement of individual phospholipid molecules. On the other side, it is present in between the phospholipid tails, and therefore the phospholipid tails on both sides of the cholesterol molecules are being pushed and packed closely. So cholesterol also hinders the close packing of phospholipids. It lowers the temperature required for the membrane to solidify. Thus, cholesterol can be thought of as a fluidity buffer for the membranes, resisting changes in membrane fluidity that can be caused by changes in temperature. So you can see here this cholesterol is performing a role of a buffer, and it is putting a check on the level of fluidity membrane will show. On one hand, it hampers the movement of the individual phospholipid molecules, thereby decreasing the fluidity. On the other hand, it is present in between the phospholipid tails and thereby decreases the temperature that causes the solidification of the phospholipid tails. You can see here plasma membrane having more unsaturated fatty acids will be fluid and permeable to a greater extent as compared to a plasma membrane having saturated fatty acids. Saturated fatty acids have straight tails, so thereby the fluidity of this membrane and the permeability of it will decrease if it is rich in straight chain fatty acids. And there the cholesterol, it pushes the phospholipid tails on both sides, but at the same time it also creates a room in between. So as compared to animals, plants have low levels of cholesterol. Rather, related steroid lipids buffer membrane fluidity in plant cells. So in plants the level of cholesterol is low, but there are related steroid lipids which act as buffers in plant cells. Membrane must be fluid to work properly. The fluidity of a membrane affects both its permeability and the ability of membrane proteins to move to where their function is needed. So there must be a proper check and balance to ensure that the fluidity of the membrane is maintained at a proper level under a given set of environmental conditions. So when the membrane solidifies, its permeability changes and enzymatic proteins in the membrane may become inactive if their activity requires movement within the membrane. So the movement of the proteins that are embedded in the plasma membrane is hampered when the plasma membrane solidifies. So the proteins, some of the proteins can move along the plasma membrane and they can rush to the site where their function is required. So for example, enzymes rush to the area where they play a role in catalyzing a reaction. So when the membrane is too much solid, then obviously this movement of proteins towards the site where they are desired is hampered. On the other hand, if membranes are too much fluid, then still it hampers the function of proteins. Therefore, extreme environments, for example those with extreme temperature, poses a challenge for life, resulting in evolutionary adaptations that include differences in membrane lipid composition. So for the proper functioning of proteins, a proper level of fluidity is also required. Now, evolution of differences in membrane lipid composition. So obviously the cell is covered by a membrane, and then we have lot of types of cells in the living kingdom, and then the organelles present inside, suspended in the cytoplasm, they are also covered by distinct specific membranes, and the membrane composition of the organelles is also different from one another and from the plasma membrane. So these variations in the cell membrane's lipid composition of many species appears to be an evolutionary adaptation. Evolutionary adaptation to what? Evolutionary adaptation to maintain the appropriate membrane fluidity under specific environmental conditions. Now, for instance, the fishes that live in extreme cold environments, their membranes will be in higher proportion of unsaturated fatty acids with the bent tails, enabling their membranes to remain fluid in spite of the low temperature. So when there is a kink in the phospholipid tails, then obviously it increases the fluidity and the permeability, because it pushes the adjoining phospholipid tails. So in such cases where the organism is living in extreme environmental conditions like extreme cold, then their membranes have higher proportion of unsaturated hydrocarbon tails. At the other extreme, when the organisms are living in extremely hot temperatures, like the bacteria living in hot springs in America, in the Yellowstone National Park, so their membranes include unusual lipids that may prevent excessive fluidity at such temperatures. So you can see here the two extreme conditions where the fishes are living in freezing waters, the bacteria are living at boiling temperatures, but their membranes are supplied with components to help these organisms survive in those conditions, and to help maintain a proper level of solutes and ions inside the cell and maintain homeostasis. On one hand, you have higher proportion of unsaturated hydrocarbon tails. On the other hand, there are unusual lipids that prevent excessive fluidity. So the ability to change the lipid composition of cell membrane in response to changing temperature has evolved in organisms that live where temperature varies. So now there is yet another condition, the organisms which are living in areas where there are fluctuations in temperature, so their plasma membrane is supplied with components which can cope with those changes. In many plants that tolerate extreme cold, such as winter wheat, the percentage of unsaturated phospholipids increases in autumn, and adjustment that keeps the membranes from solidifying during winter. So as the winter season is around the corner, the membranes are supplied with unsaturated phospholipids, so that upon the arrival of the winter season, the fluidity of membranes is maintained at a proper level. Some bacteria and archaea also exhibit different proportion of unsaturated phospholipids in their cell membranes depending on the temperature at which they are growing. So now we have come across a new concept that the organisms have the ability to manipulate the composition of plasma membrane depending upon the environmental conditions. So this is an evolutionary adaptation. Overall, natural selection has apparently favored organisms whose mix of membrane lipids ensures an appropriate level of membrane fluidity for their environment. So such organisms are favored which have the ability to manipulate the membrane lipid composition according to the changing environments. Now, membrane proteins and their functions, as you already studied that different proteins are embedded in the plasma membrane and we have so many times talked about the phrase the fluid mosaic. So the membrane is fluid because of the movement of the phospholipid molecules, and it is mosaic because the presence of a wide array of proteins. Somewhat like a tile mosaic, a membrane is a collage of different proteins. Collage means album, often clustered together in groups embedded in the fluid matrix of the lipid bilayer. So different types of proteins are embedded in the plasma membrane. More than 50 kinds of proteins have been found in the plasma membrane of RBCs. For example, phospholipids form the main fabric, but proteins determine most of the membrane's function. The membrane function is selective permeability, and this function is performed by proteins. Along with that, proteins also perform several other functions which we will study in the next two slides. Different types of cells contain different sets of membrane proteins, and the various membranes within a cell each have a unique collection of proteins. So that unique collection of proteins give characteristic properties to membranes. There are two major populations of membrane proteins. One is integral protein, which are also known as intrinsic, and the other is peripheral proteins. The integral proteins penetrate the hydrophobic interior of the lipid bilayer. The majority are transmembrane proteins which span the membrane, that is, they are present across the lipid bilayer and their ends are protrude outward on both sides. And of course, the membrane proteins are also amphipathic, so the hydrophobic portion is confined to the interior of the membrane, whereas the hydrophilic portion faces the aqueous environment both inside and outside the cell. Now the hydrophobic region of an integral protein, that is a protein which span across the plasma membrane, it consists of one or more stretches of non-polar amino acids, typically 20 to 30 amino acids in length, usually coiled into a helix. So these the hydrophobic regions, the interior region of the protein. which are present adjacent to the phospholipid tails. This portion is composed of 20 to 30 amino acids, and these amino acids are non-polar or hydrophobic. Now, the hydrophilic parts of the molecule are exposed to the aqueous solutions both inside and outside. Some proteins also have one or more hydrophilic channels. We have talked about it a while ago, through which the movement of hydrophilic substances takes place. Peripheral proteins are not embedded, they does not span across the plasma membrane, but they are usually present on the one side of the membrane, either outside or inside. On the outside, they are usually in association with lipid protein with carbohydrate portion and forms glycoproteins. And on the cytoplasmic portion, the peripheral proteins are in association with the cytoskeletal elements. So, in animal cells, membrane proteins may be attached to fibers of the extracellular matrix. Association of the membrane proteins both on the inside with the cytoskeleton and on the outside with the extracellular matrix is important so that a cell can maintain a proper shape in an aqueous environment on both sides. These attachments combine to give animal cells a stronger framework than the plasma membrane alone could provide. So, of course, plasma membrane also gives a shape to animal cell, but the association of these proteins embedded in the plasma membrane, their association with the extracellular matrix and the cytoskeleton on the outside, they play more major role in providing a proper shape to the animal cell. Now we will study different functions of membrane proteins. So a single cell may have different membrane proteins that carry out various functions, and one protein may itself carry out multiple functions. So a single protein can participate in carrying out different functions. Thus the membrane is a functional mosaic as well as a structural one. The membrane proteins makes the membrane not only a structural mosaic, but these membrane proteins play a wide array of roles, so therefore it also makes this membrane a functional mosaic. So on the right side we start studying different function of membrane proteins. Transport, of course, is a major function of the membrane proteins. The membrane proteins are amphipathic, their hydrophobic portion is confined in the inner region of the membrane, whereas hydrophilic ends are facing towards the aqueous environment. So in such a protein, how the hydrophilic molecules are going to travel? These proteins have an inner channel, and the channel is hydrophilic in nature, so therefore the transport of molecules can take place in this hydrophilic channel. And there they have discussed the consumption of ATP. Consumption of ATP is of course required when the transport is against the concentration gradient, and the carrier proteins they require ATP for the transport of materials against the concentration gradient. We have already studied that in sodium potassium pump. Now the second function of the membrane proteins is the enzymatic activity. The proteins which participate in a metabolic pathway are usually arranged together so that they can catalyze different steps of that metabolic pathway. Now another function of membrane proteins is signal transduction. There are some molecules which cannot pass through the membrane, which cannot pass through the proteins. For example, the protein hormones, they cannot invade the phospholipid bilayer. Therefore, the protein hormones, they uses a second messenger system through which they communicate the message. The first messenger or the protein attaches to the receptor on the outside, and then this information or this message is relayed by the protein towards the second messenger waiting inside the cell, and in this way this signal is transduced. Now, cell-to-cell recognition, this is of course very important because the cells must interact with their environment. The cells forming a tissue must interact with each other. The cells in the immune system must have some mechanism to identify the invading pathogens. So in order to perform this function, the glycoproteins serve as identification tags that are specifically recognized by membrane proteins of other cells. So this type of cell-cell binding is usually short-lived compared with that shown in the next slide, which is permanent. So here we will also discuss the role of carbohydrates in cell-cell recognition. Cell-cell recognition is the ability of a cell to distinguish one type of neighboring cells from another, and it is important for the proper functioning of an organism. It is important, for example, in the sorting of cells into tissues and organs in an animal embryo, because the tissue is the aggregation of similar cells. So how these similar cells will identify each other, will recognize each other, these for that purpose they need to have some kind of mechanism which helps them in recognition. It is also the basis for the rejection of foreign cells by the immune system. The cell surface receptors or markers present on the immune system cells, they identify the invading pathogens, and thereby their activity is initiated. Now, cell recognize other cells by binding to molecules often containing carbohydrates on the extracellular surface of the plasma membrane. So, the carbohydrates are attached with the proteins and that form glycoproteins. Now membrane carbohydrates are usually very short and they are branched chains of fewer than 15 sugar units, and some are covalently bonded to lipids, thereby forming glycolipids, whereas others are bound with the proteins, thereby forming the glycoproteins. The carbohydrates on the extracellular side of the plasma membrane vary from species to species among individuals of the same species, and even from one cell type to another in a single individual. So there is tremendous diversity in the carbohydrates which are attached with the phospholipid head region, and with the proteins embedded in the plasma membrane. They vary from species to species, from individual to individual in the same species, and even from cell to cell within a single individual. So the diversity of the molecules and their location on the cell's surface enable membrane carbohydrates to function as markers that distinguish one cell from another cell. So these differences in the carbohydrate composition of cells, different types of cells, is important for proper recognition. And an example is the 4 human blood types which are designated as A, B, AB and O, and they have differences in their carbohydrate portion which is attached with the proteins. So the presence of these different types of glycoproteins on the surface of the RBCs give them characteristic properties, and on the basis of differences in these glycoproteins there are 4 different blood groups. Now continuing from the previous slide, we said that the association of the glycoproteins with other receptors is temporary or short-lived, and the intracellular joining it is permanent and long-lasting, and it takes place during tissue formation. Now another function of the plasma membrane proteins is, of course, their role in providing a proper shape to the cell. They are in association with cytoskeleton on the inner side and with the extracellular matrix on the outer side, and thereby playing a role in giving a proper shape to a cell. So protein on a cell surface are important in the medical field also. For example, a protein called CD4, the CD4 stands for cluster of differentiation 4, and it is present on the surface of immune cells, and this protein, it helps the HIV virus to infect these cells, and as a result the individual acquires this disease. But there are instances where some people are repeatedly exposed to HIV and they does not acquire this condition, this disease. So it is a kind of a blessing in disguise that they have a mutation in one of the co-receptors, CCR5. CCR5 stands for CC chemokine receptor type 5. So this is a co-receptor the virus needs to attach not only with CD4 but also with CCR5 properly. Only then it will be provided entry to the cell. So when there is a mutation in the gene encoding this protein, then the virus cannot bind properly with the two receptors, and therefore the entry of the virus to the cell is prevented. So although it is a mutation, but it is an example that some of the mutations are useful. So there you can see the HIV virus must bind to CD4 receptor and CCR5 receptor, and only then it can invade into the cell. But if there is some kind of mutation in the gene encoding this receptor, then this virus cannot bind properly, and therefore it cannot gain entrance into the cell and infect it. So a person carrying such mutation is a kind of immune to this deadly virus. Now, sidedness and synthesis and sidedness of membranes. So from where all these membrane comes, where they are synthesized. Membranes have distinct inside and outside faces. The two lipid layers may differ in lipid composition, and each protein has directional orientation in the membrane. So in this diagram you will come to know how these membranes are synthesized and how they are supplied with the substances which are important for the function they perform. So in the first step, secretory proteins, membrane proteins and lipids are synthesized in the endoplasmic reticulum. If you remember, we discussed that both smooth and rough endoplasmic reticulum synthesize membranes. In the endoplasmic reticulum, the carbohydrate portion are added to the transmembrane proteins, and remember we discussed in our lecture in Golgi bodies that this carbohydrate portion will later on be manipulated in the Golgi bodies. Some of the monomers will be removed and it will be replaced with other monomers. So the synthesis of the membranes, the lipid bilayer portion and the proteins, and furthermore the membrane proteins are supplied with the carbohydrate in the plasma membrane, in the endoplasmic reticulum, and then these materials are transported in vesicles to the Golgi apparatus. What happens in the Golgi apparatus? The glycoprotein portion is modified. Some of the monomers are removed and replaced with others. And what else happens here? The carbohydrates are attached with the lipid portion. So remember, the carbohydrate is attached with the protein in endoplasmic reticulum, and the carbohydrates are attached with the lipids in the Golgi apparatus. So here glycoproteins are formed and here glycolipids are formed. Now the glycoproteins, glycolipids and secretory proteins are transported in vesicles to the plasma membrane. As these vesicles fuse with the plasma membrane, the outside face of the vesicle becomes continuous with the inside face of the plasma membrane. So here these vesicles are fused with the plasma membrane, and the secretory proteins have been delivered to the plasma membrane through the process of exocytosis, and then these glycolipids and glycoproteins are transported towards the outside face of the cell, because they are involved in recognition. Now, the biological membrane has emergent properties beyond those of many individual molecules that make it up. A membrane exhibits selective permeability, that is, it allows some substances to cross more easily than others, and this ability of the membrane is important for maintaining a balanced internal environment, homeostasis, and of course it is important for the existence of the cell. The fluid mosaic model helps explain how membranes regulate the cell's molecular traffic. In our next lectures, we will discuss different types of movements across the plasma membrane, where you will see how the constituents of these membranes ensures the prime property of the membrane, which is selective permeability. Now, consider the chemical exchanges between a muscle cell and the extracellular fluid that bathes it. Sugar, amino acids, and other nutrients enter the cell, and metabolic waste products leaves it. The cell takes in CO2, and CO2 is used in cellular respiration, and then the cell expels carbon dioxide. Also, the cell regulates a proper balance of inorganic ions by shuttling them or by sending them one way or the other across the plasma membrane. So in spite of such heavy traffic, the cell membranes are selective in their permeability. They does not allow everything to move at their own will. There is a proper control to the movement of molecules. Substances do not cross the barrier indiscriminately. The cell is able to take up some small molecules and ions and exclude others, prevent others from entering. Now, the permeability of the lipid bilayer. So as you know that plasma membrane is selectively permeable, some of the molecules which are soluble in the lipid bilayer, these molecules can pass through the plasma membrane easily. Other molecules which cannot invade the lipid bilayer, they use specific channels and transport proteins, and still other molecules cannot pass through the plasma membrane. So non-polar molecules such as hydrocarbons, carbon dioxide, and oxygen are hydrophobic, as are lipids, so they can invade and move easily across the lipid bilayer. However, the hydrophobic interior of the membrane impedes direct passage through the membrane of ions and polar molecules which are hydrophilic. So the hydrophilic molecules cannot pass through the lipid bilayer because their movement is slowed down by the interior hydrophobic portion. So what will happen? Polar molecules such as glucose and other sugar pass only slowly directly through the lipid bilayer. So therefore these molecules are transported through specific channels. We have GLUT channel for the movement of glucose molecules into the cell. Then we have aquaporins for the movement of water molecules into the cell. Then we have transport proteins for the movement of sodium and potassium across the plasma membrane. Specific ions in a variety of polar molecules can't move through cell membranes on their own, so these hydrophilic molecules can avoid contact with the lipid bilayer by seeking the passageway supplied by the transport proteins. The transport proteins span across the plasma membrane and they undergo a structural change during the process of transporting materials across the plasma membrane. There is another type of transport proteins which are called channel proteins. These channel proteins have an interior channel which is hydrophilic in nature, and therefore hydrophilic molecules can pass through these channels easily. So here you have the aquaporins, which are the channels for the transport of water molecules across the plasma membrane, and this is a computer generated model, and it shows water molecules which are red and gray passing through an aquaporine, which is a blue ribbon in a lipid bilayer. So you can see here the channel protein. There is an interior hydrophilic channel which provides passage to the hydrophilic molecules, and these are the carrier proteins where a change occurs in their shape and thereby they transport materials. So this was the end of the lecture. Thank you very much, and Allah Hafiz.